let's take a look at this. I think this was a really interesting graph. This one really, I think, is probably one of the harder graphs to deal with. The graph types, it's not the hardest. I honestly think some of the more confusing graphs are histograms. They're just annoying. But when we look at this, what we see, I don't know if you noticed this when you looked at it the first time, but there's two y-axes. There's a relative concentration of nutrients here, and there's bacterial population density, and that's cells per milliliter. And when they say concentration, I mean for every cell per milliliter of nutrient, right? Cells per milliliter of nutrient. And so you got two things going on. The nutrient concentration and the density concentration are both on the y-axis. So 10 to 1 is both this bacterial cell population density, and so this point's both the bacterial population density and the relative concentration of nutrient. So when you see a graph like this, unless they tell you there's different units, for the most part, the lines are going to mean the same thing. Sometimes they have different units on here as well. So I don't know why they would do that to you, but they will. So they have. So I look at this line. This is the line they gave you. This is the line that one of your fellow students wrote. One of your fellow students was very, very intelligent, and he found this slope. And I'll show you where. And he got this question right, and I was very happy that he did it. But let's go ahead and look at this. This is the line of cell population growth. And notice it goes up. I see it curves. It's logarithmic growth. I hope by this point in your math career you can understand that this curve is a logarithmic growth curve. And then here's another logarithmic growth curve. So it seems like there's two different places where the growth stabilizes. It doesn't decrease, right? It just stabilizes. Then they have two other lines, and they label this as bacterial population density. If I were a magazine editor and somebody turned in a magazine a graph like this, I would tell them I don't like it. I would tell them you have to be clear in your notation. I think this is relatively unclear as to what's going on. But in any case, here you can see, again, because of this y-axis, this nutrient 1 starts off high and then starts to decrease. And here this nutrient 2 stays stable, doesn't change at all, isn't increasing or decreasing, right? Until you get to about here, until what happens? When does this start to decrease? When you look at this, when does this actually start to decrease? Right, okay, good, you're looking at the number. But relative to three lines, the relationship between all three lines, nutrient 1, nutrient 2, and population density, when does nutrient 2 actually start to decrease really? When population increases the second time, good. And what about relative to nutrient 1? After nutrient 1 is gone. So these bacteria don't even start eating nutrient 2 until nutrient 1 is gone. So which do you think they prefer? They prefer nutrient 1. But you see how hard it is to read that? That's real hard to read that. So you got to be very careful, real thoughtful. So this doesn't start to get eaten until nutrient 1 is completely gone. This gets used up, nutrient 1's used, so they're growing really well. And then nutrient 1's gone, and there's some lag time, there's some wait. What do you think it's doing what do you think these bacteria are doing after nutrient 1's gone, but they haven't started growing yet? They haven't eaten any of the nutrient 2, right? They have no food yet. What do you think they're doing? You may not know this. You may not remember what a bacteria would need to do. Think of a bacteria. What does a bacteria have to do in order to switch food types? All you got to do is choose, right? I'm sick of eating bacon. I'm going to switch to carrots. That's easy, right? But this is a bacteria. It can't do that. What does it need to do? Kind of. Actually, you're not too far off, but it's not mitosis. But it's a good guess. Kind of. No, it's not dividing. The reason I said it was a good guess is because it does have to do with the nucleus. You have to turn different genes on. Because it has to, in order to switch foods, it has to switch the enzymes it's making. So if it's eating a lot of, let's say, plant material, it's going to have one set of enzymes going, and that way you can break down the cellulose. And then you got rid of the cellulose, or maybe it's eating meat. It's not eating any cellulose yet. And then it gets rid of all the meat, all the protein's gone. Now it has to switch to cellulose. So now it's going to have to switch its whole metabolism. It's going to have to switch a whole set of genes off and turn another set of genes on. And to do that, it takes time. So biologically speaking, that's what's happening here. You can't see it here in the graph, doesn't it? You'd have to, by the time, I didn't ask you to answer those questions for the record. I'm just telling you because that's the kind of question that by the end of our discussion about turning on genes and turning genes off, by the time we're done discussing metabolism and enzymes, you should be able to apply that knowledge to this situation. Yeah.
So while they were turning the genes on and off, they weren't changing the population density?
That's right. They weren't changing because they were changing their genes. Then once they change their genes, now they can eat this food, and now they can grow. They can multiply. They can they don't do mitosis, they do binary fission. Remember, they don't have a nucleus, and mitosis is the nuclear division. So they can't divide, and they can't grow and divide until they can eat this food, and they can't eat this food until they switch their genes. So they prefer nutrient one, they ate it right away, and they grew right away. Then they kind of ran out of food, so they stabilized. And they stabilize their population, they switch their genes, and they can eat food too. And that's what's happening on this graph. So they say estimate the maximum population density for the culture. That's easy. I think most people should have gotten it. Did you get your test back? Didn't I ask you to pass the test? Nobody listens to me. Okay, don't worry about it. Well, I'll give them back to you later. I wanted you guys to do, when I'm done here, I'm going to have you guys do peer feedback. So you get your test back. I already graded his. I used his as a key. And you go in, and I gave you feedback, correct? And I also posted it, by the way. I put it in your white paper, and I also gave it to the principal, just so you know, in case so you're not embarrassed. So, in any case, all of you are going to go over this with each other and give each other feedback. So I need you to find one person you trust in the room, and you're going to discuss it. You're going to give each other that paper, your paper. You can give her or him your paper. You're going to take theirs, and you're going to look it over after I give you all the answers, and then you're going to give them feedback. Is that clear? All right. So you estimate the maximum population for the culture. I mean, I think that's easy. It's 10 to the 8. All right? Everybody got 10 to the 8? If you didn't get 10 to the 8, it's the highest, like, population density. It's the highest one. I mean, does that make sense? Is anybody confused about that? I know maybe I shouldn't have said it was easy, because then you might feel... It's just you look at the graph and you just see where it's the highest. So that one should have been easy. And it says cells per milliliter. That's really great. This is why having units and being able to read units it makes your life easier. I can look at cells per milliliter, so that's definitely what I want to look at. All right. Describe what prevents further growth of bacteria. After that, what do you think happened? Why can't it grow any further? There's no nutrients, no food. It's out of food. See? Nutrient 1's gone here, nutrient 2's gone here, so there's no more food. I didn't ask you to answer that, but that's an easy one, right? But you'd have to understand what was going on in order to answer it. All right, so using the data, calculate the growth rate in cells per milliliter of the bacterial population between 2 and 4. This is where this student did really well. He picked 2, and then he picked 4, correct? And he knew, he remembered this one thing, that rate is slope, and slope is rate, right? So between 2 and 4, the rate is a straight line. That's beautiful. That's why they pick 2 and 4, by the way. It's a straight line. So I pick 1, 2 points on that straight line. I calculate the slope, and that's my rate of growth. It's like 4,000, something like that. All right. Does everybody understand how to do that? Everybody remember how to do slope? Okay. I mean, I know it's to some. Hey, I have a bunch of ninth graders can't do average, man. I have to go over it with them. It's okay. I forget stuff. I misspell things, obviously. So, okay. Thank you. Thank you for that. Appreciate it. Appreciate it. Thank you. Your mercy will be remembered on when I grade your essay. Thank you. All right, identify the preferred nutrient of the bacteria. I didn't ask you to do this, but I think right now you could probably answer nutrient 1. And when they say justify, you would say something like nutrient 1 was used up first to grow, and it took time for them to switch their genes over to eat the second nutrient. All right, so describe how nutrient 1 most likely regulates the genes and metabolism in nutrient 1 and genes and metabolism in nutrient 2. This one I'm not even going to get into until we talk about gene regulation. So I think that's... So let's think about this. If you took the AP exam today, without going over most of this biology, correct? And this is the FRQ. This is 40% of your test. If you took it today, you would have got half of this question right, probably. Maybe 3/4 right. 3/4 of the points without going over any of that biology. Just by reading a graph and being able to read the units, that's not bad, right? All right. So again, this graph, no reason to make it so utterly confusing, I think. It's like, what am I looking at? But this person also got this right, or mostly right, by the way. This person, without going over, what did we cover in the book? Like 3 chapters, basic stuff from chemistry. This person would have passed their AP exam in 2016. They would have, assuming they had a decent score on their multiple choice, they would have scored 3 or 4 on an AP exam without going over the biology, just because they could read graphs. So you got, these are the seeds, these are receptacles, these are flowers, these are leaves, these are vegetative buds, these are stems, these are roots. And the colors are there, and this is percent dry weight, and this is time in weeks, obviously May, June. So if a plant is growing in April, May versus June, July, what's the difference between May, June, July, and August? What is the difference? Yeah. Oh my God, this Siri thing. She's still talking when I close the computer. All right. What is the difference between the months? Yeah. Temperature's good, but oh my God. All right, another good one, correct. But is humidity, rainfall, temperature, all correct. Come on, guys. What's the difference between June, July, August, April, January, February?
They come after, yeah, they're numbered differently.
Okay, the months are numbered differently. All right.
The number of dry, percent of dry weight, like from the different types of sources.
Okay, but my question is, what's the difference if a plant is growing a plant?
Oh, it's using more of a certain.
A plant. Photosynthesis is going to be different. Why? The amount of sunlight. Come on, we're just coming off the summer where we had like 9 hours of daylight, right? 10 hours, right? Didn't we have like something like 9 hours a day? And now we're going home, and now it's getting dark at like 6, 7 o'clock. It wasn't getting dark until like 8 or 9 a few months ago. So amount of sunlight. Okay, it may not have been obvious, but that's something to think about. That's the source of energy for plants, is the sun. But how does a seed grow in April? The sunlight is minimal. What would you expect it to depend on? It's leaves? Does a tree have leaves in early April, in March? Not if it's deciduous, it doesn't have leaves yet. They just start. The soil is not the answer. Something in the soil, roots. That's where plants put their energies in roots. Now, do you know all this? No, we haven't talked about it. You haven't read it yet. So we talked about it in ninth grade, but that was a long time ago. Okay, fine. So they're in the roots. You know this because you potatoes are roots, don't you? You know yams are roots, right? Did you know that? You know carrots are roots? You didn't know that? These are all roots. And this is where can we close those computers and pay attention to what's going on? So the roots, close those iPads as well, please. Close them. Thank you. Okay, then just close it. If you're not on it, doesn't matter then. All right, so I would expect, if I was looking at this graph, before I even try to analyze this crazy graph, if I'm looking at all these different things, I would expect that most of the energy is going to be coming from a seed or a root in the early months, and come around middle of summer, I'd expect all the trees, all the plants to have leaves, and they'd be doing a lot of photosynthesis. That's what I'm expecting before I even look at this. All right. Now I'm looking at it, and it says, from May to late August, the percent dry weight can be used to estimate the amount of energy plants uses to produce its leaves, vegetative buds, stems, roots, reproductive parts, seeds, receptacles, and flowers. Identify the direct source of energy used for plant growth during the first week of May. So here's May. There's May. First week of May. What is the direct source of energy? Look at the roots. It's just reading the graph at this point. Roots, right? It's going to use its roots because there's no leaves. It needs to build the leaves. It needs energy. So it's this energy the plant stored last summer for the winter.
It's its direct source, so wouldn't that be the sun?
It's not using the sun. Why is it not using the sun? It doesn't have leaves, right? But you're right. You're right. It would be the sun if it had leaves and it was a plant. In fact, where do we see what's happening in May or June? Where's most of the energy coming from during these months, June and July? The leaves, which would be the sun, right? Sun, leaves, roots, stored energy. All right. Based on the data on the graph, estimate the percent of total energy that... Now, by the way, to answer the first A, you did not have to remember roots. All you had to do was read the graph. I just want to point that out, right? That's how this kid got it right, or maybe they knew roots was... But the bottom line is that you could read it without... You can answer A without remembering everything I just talked about. You don't have to understand the biology to read this graph. Just identify the shade. Roots, roots. Okay. Based on the data on the graph, estimate the percent of total energy the plant has allocated to the growth. The leaves of the final day of July. Here's the final day of July. What does it say? Oh, the first day of July, sorry. First day of July. And the percent of total energy that the plant has allocated to growth of leaves. All right, so there's first day of July, leaves. So somewhere between what? I would say 50. Oh, that's June. That's July. First day of July. June's over here. May's here. First day of May, first day of June, first day of July, right? Is that correct? Do you agree?
Yes.
Wait, am I wrong? This is the first day of July here. This is the first day of June here. This is the first day of May here. Do you see that?
Yeah. Oh, the increment. Yeah.
All right. And this is the first day of August. So first day of July, I would say leaves about 50%. About 50%, somewhere around here. It's hard to read this, to be honest. I look at this and I'm like, what is it 50? Or is it 80? Is it from 50 to 80? Depending? Like, how is that an answer, right? But the answer from College Board is 50. And I guess you can say 50, but then again, why isn't it 30? Well, that can't be 30 because this is leaves. So it could be anywhere from 50 to 80. So I would say 50 because it's the first thing that I get to. I said 50 because it came up here and I saw 50 first. But then I thought, well, what if it's 80? Just pick 50. Pick the first one, 50. I think they'll let you get away with 50 to 80. But I don't know. I'd have to figure out how to read this graph. It's really crazy. Okay. Then, and it says, it does, to be fair, it does say estimate. It doesn't say exact. Compared to perennials, plants that live more than 2 years. Perennials live more than 2 years, by the way. Annuals only live for a year, which I think is kind of silly to call it annuals because I just always think like they come back every year, but that's not right. Annual plants often allocate a much greater percentage of total energy growth to their reproductive parts for any given year. Propose one evolutionary advantage of energy allocation strategy to annual plants compared to that with perennial plants. That we're not going to discuss. I mean, you didn't have to answer it, but I just want to point out that, look, if you're an annual and you spend the entire year, you don't have to put any I was going to say money, but it's any energy into root development, into stem development. Only thing you got to do is collect all that energy, make some seeds, and then you can die. Spread your seeds and you win. That perennial has to build its stem, its structure. It has to fight off disease. I mean, it has to live year after year after year, and it's going to have to develop enough food source to survive the winter as well. That's a lot of effort. Where the annuals can quickly grab as much energy as you can to fill up a seed, or as many seeds as you can, and then die, and you win. So that's two different strategies. They're both good. Depends on your view of good, but annuals are really only mean to live 1 year, and they have no problem with it. For the record, there are things around here that would be called annuals that are actually perennials in other areas. It's just that our winter wouldn't let them live from year to year, but irrelevant. That's the point. Everybody okay with that? All right, so that's that question. Again, I hear you probably get a little less because there's a little more biology, especially one evolutionary question. But really, two out of 3 questions is just simply read the graph. All right, let's look at the next one. All right, so here, construct a graph, and it gets all kinds of crazy, like LAP are enzymes that remove nitrogen terminus of the amino acid, and there's populations of M. edulis. Researchers can sample the adult mussels in several locations, and they gave you a graph. It's from 1 to 8, right? I don't know if you saw that. I have it here, but I'm not going to pull it up. The Long Island Sound is from 1 to 8, and the Atlantic Ocean is here. These 3, 6, 7, and 8 here were Atlantic Ocean. Long Island Sound was up here 1 through 5. Actually, I think I should bring it up, but because it's about reading a graph and reading a I don't know if there it is. It's reading a graph and being able to read a map. All right, so when you look here, this is you all know that this is the ocean here, correct? Do you know this is the ocean?
Yeah.
Like, can you tell when you read here? It says Atlantic Ocean. Okay, what does it say? High salinity. And this is increasing salinity. This kid actually got it wrong, this question wrong, even though he graphed it well. You'll notice all the checks here. Because he didn't read this, I'm assuming. He didn't read increasing salinity, so you go from 1 to 5. This is the max salinity here, and here these are all pretty much the same salt concentration. Why would it increase from here to here? Why would it increase from salt concentration from 1 to 8? What's happening from the land when it rains? Fresh water goes in the ocean. So this is a mixture of fresh and salt water. So it's diluted, and when it gets closer to the ocean, then it becomes more stable. So everything's moving towards, all water that falls on the land ultimately theoretically ends up in the ocean, unless it gets trapped in an aquifer. So as you see here, you're going to have decrease and increase in salinity this way. And here, all these are going to have the same salt concentration, maybe a little less if it's really next to the shore, because you're going to have some fresh water getting dumped there. All right, so we look at this and we graph it. We see different locations. And we see here individuals with the gene. In the salt water, they're all the same. But in areas 1 through 5, where you have an increasing salinity, you see the gene is expressed more in the more salty areas. And in the 3 that are in the Atlantic Ocean, which have all the highest salinity, they all have the highest expression rate at about 60. Do you all agree? Yes? Do you all read that? This is the graph this young man made, and he messed up the answer to the question only because he misread what he was looking at, not because it took a lot of understanding of biology. He got points for these. Each of these checks is a point on the FRQ. Putting the units, making the correct label. He lost points because he didn't label the graph. You have to actually give it a title. If you don't give it a title, you're going to lose points, I'm telling you. He got points because he had a key. So simple graphing skills will get you points on that FRQ. That's why we went through all this graphing and statistics. Because literally half of your FRQ, and I would argue probably in a lot of your AP exams, is about graphing and graph interpretation. Yeah?
So what could they have done differently?
They misread this. They got the graph right, except for the title. They have mostly right. Where they had it wrong, where they got wrong is they messed up. They didn't read increasing salinity from 1 to 8.5. They didn't read that. So it was this that they got wrong, that they messed up on. And you see, this says high. Why is the Atlantic Ocean all just high? Because it's all the same concentration. It's so big, it's hard to change the salt concentration. We are changing the salt concentration in the ocean, but we're doing it because we're melting the ice caps, which are mountains of ice, fresh water. They're sitting on top of Antarctica. Those are melting, and because they're melting, yes, we are diluting the ocean. But that's a lot of water. This amount of water in this sound is not going to impact the salinity of the entire ocean. Did you all get that, or are you busy looking at your navel?
What?
What's he doing? You've never heard of navel gazing? There are people that gaze at their navel. I don't know why. All right, so what are you doing? You're not focused on what I'm pointing to. I'm pointing to stuff on the board. Your back is to the board.
I think it would
Huh? Yeah, I wanted to pass it out, but certain people didn't listen to me when I said pass it out. All right, so I'm going to let you pass out. You guys give each other feedback, and if you need me to explain, come and get a question answered from me. Yeah. Oh wait, I need to listen to Stratford's question or answer. Go ahead. What?
I'm saying, like, what could they have done, like, wrong differently? Because you said, like, something you saw on the way they put graphics, you know, that they didn't read that part.
Oh, how did I know that they didn't read that part? Because they had a question. The question they answered was reversed. They thought, I forget what the question was, but I wrote it on their answer sheet. You have to read the question. I can't remember. I read this 2 days ago and I graded it. I don't remember what it said, but it said I don't remember. But they reversed this 1 through 8, and it tells you increasing salinity.
So it should have been, like, smallest to biggest?
It's smallest to biggest. That's what increasing means, right? You start small and you get bigger. No, the graph is right. The graph is right. There's nothing wrong with the graph. There's nothing wrong with the graph. It's just when he answered the question, he got it wrong because he didn't see 1 to 8 is increasing. And maybe and honestly, Yeah, I guess. Because here is not, this is not increasing salinity. Here's increasing individuals with that particular gene expressed, or with that particular gene, increased with the increase in salinity. But he thought it was increasing with the decrease in salinity. So he just mixed this, because you had to combine both charts to really interpret the question and get the answer right. So even though this is just a bar graph, there's a whole another level here of depth where you have to apply that bar graph to this specific situation. All right, so I'll post this, and then you guys can have a discussion. And if you have any problems or conflicts with your discussion, your feedback, let me know.